All right, so cells and tissues are obviously impacted by signals, and these could be local signals, like maybe, someone in this train is telling someone else to kind of move over politely so they can get by. There's also physical forces, like maybe the people hanging on for dear life in this structure, and then geometric constraints, right? In this case, these people are all packed into a finite amount of space. So all these things are important considerations when thinking about tissues. So in tissues, in order to change shape, there's a sequence where forces are transmitted from the molecular level, on which the signals also operate on, up to the cell level. And I'll just show you, this is my kind of favorite signaling pathway and favorite structural proteins, which is Rho kinase and actomyosin. So Rho kinase here would be a signal. It's downstream of the GTPase Rho A. And then the molecular motors and force-generating machinery, myosin and F-actin. So in this case, right, these molecular level structures are going to organize at the cell level and generate force to change cell and tissue shape. Right? In this case, myosin assembles in the middle of this cell to contract it. Okay, so this is the basic sort of structural unit that I'll be showing you in my talk. But these cell forces, right, in order to change the tissue shape, have to transmit forces across between cells and across the tissue. And for that, the adherence junctions or some junction type are important. And it's required that these motor cytoskeletal structures are coupled at adherence junctions so that forces can be transmitted in chain sort of across from cell to cell. Okay. Now, what's really fascinating about this process is tissue level forces feed back on these other sort of smaller structures in order to influence the overall behavior of the system. Okay, so as Thomas Lequi showed you earlier in the meeting, right, there's kind of all these feedback loops that go into dictating what the final shape of the structure will be. So for today, I wanted to tell you a couple stories. First, I want to tell you about how geometry affects how cells interpret signals. Then I'll go and tell you about how signals sort of add into sort of tissue level forces and some important principles there. And then finally, I'll talk about how the forces and the cytoskeletal structures that assemble in response to the signals then feed back and influence the signaling in the cell. And I want to encourage, if you have questions during my talk, please feel free to interrupt. If I have to drop something, I am prepared to do it. I also try not to stuff it so that there's time for discussion. All right, I'm going to use, or my lab has used the fruit fly embryo and specifically the gastrulation in the fruit fly to get at some of these fundamental principles. So I'm just going to give you a brief introduction to the fruit fly. So this is a Drosophila embryo in the top left there. This is an ellipsoidal shaped embryo, kind of like an American football. And at this stage, which is about 2.5 hours into development, you can see that the surface is relatively smooth. And what happens, or what will happen with this embryo, or an embryo like that, is that the ventral side of the embryo, the ventral cells, internalize in a process known as gastrulation. Okay, and this results in this seam along the ventral midline. And the surrounding cells, these blue cells, which start out on the lateral flanks, come together and meet at the midline to close off the mesoderm from the surface. Okay, and so this is shown in cross-section here, where you can see the presumptive mesoderm labeled in the black there. That's a twist stain, which marks the mesoderm. You can see over time, this area at the bottom starts to change shape And eventually these twist positive cells internalize, forming this internal tube, which will eventually dissociate and form an inner layer known as the mesoderm. Okay, so this is the process. Now this process of gastrulation involves different cell behaviors occurring across the entire embryo. Okay, and these behaviors are regulated by discrete sort of gene expression patterns, which are well characterized in the fly. But I want to really emphasize that it's the cooperation of all these cell behaviors which is contributing to this dramatic kind of mesoderm internalization. So as I mentioned, the mesoderm is on the ventral side of the embryo, shown here in red. And it's a cell shape change, apical constriction, which initiates the process of mesoderm invagination. In addition to this apical constriction, there's tissue right next to the mesoderm, flanking the mesoderm, that around the same time, the cells in that tissue are going to start undergoing migration, and they're going to intercalate between each other. And this is important for sort of driving convergence of the tissue and sort of ventral directed movement of the more dorsal part of the tissue. And also it initiates the body elongation of this animal. Okay, so this is sort of tightly coupled to the internalization of the mesoderm. And finally, in the back of the head, cells undergo mitotic entry, and they undergo divisions, and that also accompanies this process. So I'll touch on all of those sort of over the course of my talk. All right, so the first story I want to tell you about is how does geometry, how does that affect how cells interpret signals? I'm going to tell you about work done by Céline Chanet, who's a former postdoc in my lab, who's now a permanent researcher in France. And her story is that she showed how the packing of cells in an epithelium is actually important for cell division orientation. And then I'll tell you about a story from another former postdoc in my lab, Nat Clark, who showed how mechanics and signaling sort of together coordinate this internalization of mesoderm with the body axis extension of the embryo. So for cell division, the orientation of cell division is extremely important for morphogenesis, and it's influenced by both molecular and geometric cues. The molecular cues that are involved are cortical proteins such as pins and mud, which recruit the microtubule motor dynein, which by pulling on astral microtubules rotates the spindle such that the spindle and the cell division acquire a particular axis. But there are also geometrical cues, such as cell shape. And cells tend to divide along their long axis. This is known as Hertwig's rule. And you can see that here. This cell is slightly anisotropic, being longer along this axis, and the cell divides along this axis. So these two principles both operate for the division sort of for angle. We were interested in our epithelial tissue, and I'll start with the conclusion. What Céline found is that myosin-induced rounding of mitotic cells was actually critical for cells to interpret a molecular cue that orients the division in the plane of the epithelium. Okay, so I'll show you the mitotic domains here. So normally in Drosophila gastrulation, when cells divide, they divide in the plane of the epithelium. So this is 3 cells. The plane of the epithelium would be going this way, and the cells divide within this plane here. You'll see the mitotic cells round up right here. There's the mitotic domain 1. You see them round up, and then you see the cytokinetic rings divide the cell. And you see, because you're not seeing like a nice ring that's isotropic, it's because the rings are oriented perpendicular to you. So you're seeing a band. And I'll play that once again, just so you can see it. So that's showing you that the divisions are happening in the plane of the epithelium. Yes, Vanessa.
Yeah, silly question.
Mhm.
Do you know if the schematic is correct? Do the cells actually not go under to compensate when there is rounding of their neighbors? Or do you think they really stay columnar and then there is some?
I will show you.
Okay.
I will show you what the shape looks like. Yes. All right, so that's what the divisions look like. Oop. And I want to say that pins is required for the division and the epithelial plane. I'm not going to show you the data, but if you make a pins mutant, now the cells, rather than dividing in plane, will flip up and divide in the wrong way, right? And then you do get cells that when they divide, they kind of go out of the epithelial layer. Yes, Marty.
Is that because the cells are very columnar, and so then I guess the long axis is that a defect in their rounding, or is that because they are more columnar, I guess?
I will show you. Yes. So we got into this project because we discovered that myosin is actually required for this orientation. So I'm going to show you some rows plots here where if a low angle here means that it's dividing in plane, and a 90-degree angle means it's dividing out of plane. Okay, so this is data from a normal embryo showing you that the divisions are happening in plane for just about every cell that divides. Now, what we found is when we inhibit myosin, here with Rho kinase inhibitor, but also by depleting the myosin heavy chain, or by making these mutants in the light chain, and these are mutants that we've shown reduce the ATPase activity of the motor, and therefore they're going to contract more slowly and less robustly. Okay, so in each of these cases, you see a significant increase in the number of out-of-plane divisions. Okay, so myosin's required for this. So the first thing we tested is, does this affect the pins localization? And here, pins is stained in red, and you can see it localizing to the pins localizes to the lateral sides of the cell. That's thought to be why the spindle rotates into plane. And you can see in each of these mutants over here, you still have that lateral pins. So it doesn't look like myosin is required for the cue to be present, but that there's some other issue that's involved. So all of you are asking about cell shape. So what we did was to collaborate with Zia Khan's lab and basically reconstruct the shapes of these cells as they divide. So up here is a wild type cell. You can see before mitosis, it's columnar. And when it enters mitosis, you can see how this thing rounds up. It comes up basally, and it has a much more cuboidal shape. And in this case, then it divides, and both of the daughter cells are in plane. When we inhibit myosin, either with ROCK inhibitor injection or with the myosin mutant, what happens is the cell has entered mitosis, but the cell remains columnar. It can no longer round up. So it's stuck in this crowded tissue. Without the force-generating myosin, it can't round up. And when it doesn't round up, it then divides out of plane. So we think in this case, we do, as Marty was suggesting, there's a long axis here, and the long axis is sort of out of plane. And so the spindle either can't fit by rotating, or it is just sort of, it's overridden by cell shape, the molecular cue. Yeah.
Do they reorient to be parallel to each other afterwards, or do you start getting two layers?
I've always wanted to look at this, but we haven't looked to see what happens to those cells. At least over the short time that we've imaged after the division, we do not see them reintegrating. So I think the daughter cells are basically left on the basal side. Yeah, Pranati.
Relax, but along with this cell, with the tissue, still follow this group, or will it be random if the neighboring cells are not exerting any pressure or tension?
So you're asking if we can basically like release the pressure and see if they can then flip out. Hard to say. I think we have done that. We've done a lot of laser ablations. You might have noticed the divisions, the planar divisions are actually oriented. And the orientation appears to depend on sort of tension in the tissue. So it's a complicated problem because you have planar polarized divisions. The tension's required for the planar polarity. And actually pins is planar polarized. It's not around the entire lateral cortex. So in that case, they, I believe, still orient. If you release tension, they are still planar, but then they're no longer planar polarized. They divide any sort of random angle in the plane. This is just showing you how the aspect ratio, as I said here, is increased when you don't have the mitotic rounding at the time of metaphase. So it appears that here, the sort of the cell shape, the long axis rule is overriding the pin skew. All right, so the conclusion here is that mitotic cell rounding is required for these cells to actually interpret a signal that's telling them sort of in which axis to divide, right? And we think that's because of the crowding, that the cells, if they don't have this sort of energy to sort of push their neighbors away, then they're basically not able to use that molecular cue to orient. Right, so the mitotic rounding is kind of like a way for the cells to kind of make some space for themselves, such that then they can sort of follow the molecular cue. Yes, Sean?
On the cell rounding, the negative space that's created, does that get taken up by the next deeper layer of cells, or does it stay sort of unoccupied?
There's no deeper layer of cells, it's just yolk. So yeah, so the yolk probably kind of comes up when the cell. Yeah, Johan.
Did you try to rescue the phenotype by because you somehow your work this will imply that the size of the spindle
Yes.
is actually need to match basically the size of the base. So I was wondering whether you have tried to, for example, use some mutant that will reduce the size of the spindle, such as mini spindle, to rescue the phenotype and see whether in this case the cell will be able to read the cue, because you have, it seems that you have two constraints. Basically, you have the pins that seems to indicate the cell actually planar. At the same time, the spindle cannot be in the plane because of the global tissue constraint.
Yeah.
I was wondering whether reducing the size of the spindle can rescue this phenotype.
Yeah, we would love to be able to do that. So you said is mini spindle is a mutant?
Yes.
Or
yeah, mini spindle is a mutant that make very tiny spindle basically.
do you know if it's fertile? Like, can I get embryos that have it?
Yes. No, I don't know. But yes, I get because it must have been no, because it was no. This I don't.
Yeah, this is something we're still interested in, because we actually we're now looking at how these divisions are affected by sort of the mesoderm. And we would love to sort of image the spindle more carefully in some of these cases. And also we've been batting around this idea for like a decade. Like, could we shrink the spindle and show that actually Rebecca Heald was the first one who was trying to get me to do that. So I would love to be able to do that. I'll have to look into that. Yeah.
So when they round up,
Yep.
they still maintain that long axis along the AP axis, which is what allows them to sort of consistently divide, along that axis. But so if you think that's because of tissue tension, if you take embryos that have like the more roundish embryos, mutants, in that case, like, do you see this the angles change when you do when they round up?
We have not looked at the angles in a round mutant, but we could do it. It's a great idea. So, yeah, we're thinking of that as well. Okay. All right. I'll move on to the next part. So now I'm going to zoom you out, and we're going to start thinking more globally about how these divisions and other events kind of fit into the embryo. And this is a movie from Matteo Rausi and Maria Leptin. And this is going to be a movie it's a light sheet movie. And the the cylindrical embryo has been unwrapped. So the dorsal midline will be here and here. So basically you can imagine this kind of wrapped up into sort of something that resembles an ellipsoid. And you're going to see the different behaviors that happen here in the flows of tissue. The cell divisions I just told you about, the rounding happens, starts to happen at the sort of right there. But the mesoderm internalization is happening on the very ventral side of the embryo. It's been shown that the surface area that is lost by mesoderm internalization is actually in large part made up by these cells sort of rounding up and increasing the surface area of the embryo. Although a functional role for that rounding has not yet been shown. But in addition to these two behaviors, between them, there are cells of what's known as the germ band, where cells are going to migrate between each other and intercalate. And this both causes a flow ventrally, but also causes the initiation of axis elongation in the embryo. Okay, so all of these things are coordinated. Yes?
That's not right. What's this ring that's contracting, this white ring? And then the other video there.
Which ring?
There's like
This one here?
Yeah.
That's so the embryo develops as a syncytium. So that ring is the cells forming. So the membranes are kind of moving centripetally inward. That forms the epithelium. And then as soon as the epithelium, even before the epithelium has finished forming, these gastrulation movements have started. So all these processes are happening kind of like bang, 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 all together. And so we've been interested in it because we want to see how they're coordinated with each other. Oh, that's too far. I'll tell you briefly about a story that we recently published. This is work done by Nat Clark in the lab. And this shows how there's a biochemical signal, EGF, and a mechanical signal stretching by the mesoderm. And this appears to serve as an AND gate to stimulate sort of the myosin cable assembly in the germ band. So both the biochemical and mechanical signal are required together to sort of trigger this event. And this would then coordinate the sort of internalization of the mesoderm, the tissue folding, with the axis extension of the embryo. So what he showed, so here in this diagram, the mesoderm is in yellow here on the basal side, and the extending tissue, the germ band, is in magenta. And what he showed is that if you disrupt, there's also EGF signaling in the germ band, and we showed that EGF is required for the mechanical sort of effect of the mesoderm pulling the germ band to stimulate the myosin cables. And I just want to highlight a study that preceded ours by Hannah Gustafsson and Sebastian Streichen, who showed that the germ band cells are responding to pull and assembling myosin. And what we showed is that it depends on this EGF signal. So it's kind of an AND gate in terms of controlling where and when this happens in the embryo. So EGF is patterned in the embryo by the expression of this rhomboid protease, which cleaves a membrane-tethered EGF ligand, which releases the EGF so that it can signal through its receptor. And the way that what this looks like, kind of with cellular resolution, what I'm showing you here, this is a lateral flank of an embryo. It's, normally it would be sort of a circle like this. We've basically taken one side and linearized it so that you can see from the ventral midline there to the dorsal midline. So it's kind of like the sheet I showed you before, but this is just a single sort of, we can make a one-dimensional sort of view of the dorsal ventral axis here. So SNAL, the mesoderm, is in yellow here. You can see it here. There's a sharp boundary with the ectoderm. The ectoderm cells immediately adjacent to the mesoderm express rhomboid, this protease. And you can see how the rhomboid protein and the downstream phospho-MAP kinase signaling is activated in these ectoderm cells. And what Nat showed is that this EGF signaling is required to assemble sort of more intense adherence junctions, which you can see as those red spots there, and also myosin II cables in these cells at the time of the mesoderm internalization. So what's the role of force in this process? So the way we showed this is we wanted to show that it wasn't affecting EGF signaling. So what we did is to take an embryo, overexpress a constitutively active EGFR, and that leads to really intense myosin in the germ band. You're looking at the germ band here. The ventral furrow is pulling down here. You can see when it pulls, you get all this myosin appearing in the germ band. And it's actually more myosin than normal because we've activated the EGFR. However, now if you focus over here, what we've done is essentially a mechanical epistasis experiment. So we're activating EGFR. So biochemically, it should be assembling lots of myosin. But now we're going to knock out the apical constriction that's causing the internalization of the mesoderm. So there's no longer a mechanical pull. Okay? And when we do this, if you compare this to over here, what you'll notice is that there's a lot less myosin assembly. It eventually assembles, and it will eventually extend its germ band. It's no longer linked to the internalization of the mesoderm. And this is quantified down here for both control and constitutively active EGFR. So you can see when there's pulling, there's assembly of myosin. When we knock out the pulling, there's lower assembly in both cases. So even if you activate the biochemical signal, it's not enough. You require both biochemistry and mechanics. So I've shown you how geometry and mechanics can really gate these signaling responses. Yes, Marty?
I guess, so in the mutant, it would be like, that would be affecting, I guess, like, ROGEF2 signaling. And
yes.
So it would also impact myosin contractility, but it would just be apical constriction versus the junctional myosin?
Yeah, the mutant we're using, we're knocking out genes that are only active in the mesoderm. So we're basically knocking these are
there's still ectoderm myosin.
There's still ectoderm myosin, right? So we knocked out the targets of twist. We knocked out FOG and T48, and if you knock those two out, you get you don't get apical constriction, and they're not expressed in the ectoderm. That's the nice thing about Drosophila genetics here. We can really kind of target specific genes that are expressed in particular domains. Yeah, Johan.
Because the first slide that you showed us is that globally there is a higher level of E-calorie in the region where EGFR was activated. Yeah.
Yeah.
I was wondering whether the effect is of EGFR is a transcriptional effect, and then you get the localization at the apex by the forces. Or do you think that there is a direct role in controlling the level of myosin, or you think it's just the activity? Yes, it's hard to see whether it's a level effect or you see because Rho, for example, seems to
rhomboid.
Yeah, rhomboid seems to be higher in this region, and I have the feeling that there is more calorie overall, not only more calorie localized apically. The calorie, so I was wondering whether in the experiment that you show where you do the epistasis, whether the overall level of myosin is the same in those two contexts, basically.
I don't think there is a transcriptional increase in cadherin here. We think it is just kind of relocalizing or localizing differently.
So if you block MAP kinase activity, you still get this effect?
We still have to do that. We're not sure where on the EGFR pathway, and this is actually what we're actively working on in the lab, right? It could be SARC, it could be the EGFR itself. There's a lot of potential connections. So we're now screening for which of these could be directly regulating the junctions and also the myosin.
But you think the effect is via E-calorie?
It's very hard to parse. that's the first thing we see. If we knock out the cytoskeleton, we do see that E-cadherin is still enriched in that domain. So we don't think the cytoskeleton is required for the junctions. But obviously junctions in actomyosin are basically one big feedback loop. So we can't say that there's not some sort of dependence on each other. Yes, Vanessa?
Force activates the receptors?
Well, we don't know if it activates the receptors or if it's the receptors are required and sort of the pulling forces kind of act downstream. And this is what we're trying to distinguish now. Right, it could be EGFR activates something that's mechanosensitive or, yeah, so we don't know. I think we haven't looked carefully also. We're trying to get some like good EGFR activity reporters so we can really look to see if we knock out folding, do we affect the activity of EGFR? But so far we haven't seen that that's the case. They seem to be kind of independent inputs, which makes sense because this is all transcriptional. So we don't necessarily think that's a sort of mechanics dependent input.
There are many cases which signaling receptors and signaling cascade can
Yes,
be affected
yeah,
directly,
yeah,
right?
exactly.
But
Mhm.
broader sense, account sense.
Yes. Yeah, no, I think it's a great point and we will try to figure it out. Zoom ahead here. So I next wanted to talk about how do these, a lot of these signals are very dynamic. So I want to talk about how dynamic signals sort of lead to tissue level forces. And I just wanted to point out sort of a phenomenon that's been observed in many of these systems. I think I first saw it in work from Ed Monroe in the C. elegans single cell embryo, where there are pulses and sort of flows of actomyosin, which are in this case, I'll play the movie again, are involved in the polarization of the zygote. But in other systems, including the Drosophila embryo over here, we saw that these sort of actomyosin pulsings involved in epithelial morphogenesis. I'll tell you why I think that's important next. But also here in the starfish, these rho waves have been characterized in, this is a movie from Bill Bement's lab, but also in the lab of Nikta Fakhri. So you get these really striking sort of pulses and waves of actomyosin. And this is because Rho A activity appears to exhibit this excitable behavior, which I'll touch on in a bit. So what are these pulses doing in the embryo? So I'm going to show you a movie by Mimi Z, a former graduate student in my lab, just to give you a sense of how these pulses are interacting during mesoderm invagination. And what you see are these flashes. When the cell lights up, it's one of these myosin pulses. And you can see there's a very heterogeneous behavior across the embryo. They're not all doing it together, but it's almost like this stochastic salt and pepper contract, 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 contract. And so we wanted to understand why the cells are doing this in this case. I want to point out that an important aspect of transmitting or changing the tissue shape is transmitting the forces between cells. These forces are transmitted across junction structures, such as these adherence junctions. And in this case, to transmit force, it requires that there be some amount of force balance between the cells so that the force can propagate from one cell to the next. And what I'll tell you is that this force balance, what's important is the dynamics of the cytoskeleton and also the signaling. This is what our current sort of theory is for the importance of the pulsing. And we got into this by doing, starting out by doing some genetics. And we started screening for cytoskeletal genes and signaling pathways that were required for mesoderm invagination. These are genes that weren't picked up in the sort of forward genetic screens because they all are lethal in the embryo. You don't get an adult, but we're able to maternally knock down them and find ones that were important. One of the first ones we discovered was myosin phosphatase, which Claudia Vasquez in my lab and Akankshi Munjal and Thomas Lequis's lab both showed was really essential for these dynamic oscillations of myosin. So myosin phosphatase inactivates myosin and Rho kinase activates. So you get this kind of turnover loop here. My lab in a screen found this Rho gap. And so this Rho GTPase, right, can cycle between active and inactive forms. The RhoGAF2 activates it by exchanging the GDP for GTP. This gap, Cumberland gap, inactivates it by stimulating GTP hydrolysis. And we showed this gap, even though it's inactivating the signal that leads to myosin activation, is essential for furrow invagination. And what I'll tell you about today is also the actin turnover, right? Actin's a biopolymer. It undergoes cycles of assembly and disassembly. We showed that both rapid assembly and rapid disassembly are required for this process. And I'll show you what the result is when you don't have this here. So if there's a lack of force balance at this adherence junction, what you're going to see is that it's going to be pulled in one way, but there's no counter force the other way. And this results in this cell contracting. And what happens is the other cell actually gets a membrane protrusion pulled out. So there's a loss of force balance and you no longer have force connecting these two cells as effectively. So what we found is that if we decrease the rate of F-actin assembly or disassembly by knocking down genes that are involved in these processes, or by injecting cytoskeletal inhibitors that do similar things, we get a certain phenotype. I'll also say if you knock down the Rho gap or if you knock down the myosin phosphatase, you also get examples of what I'm going to show you today. So I'm going to show you the myosin channel up here, the membrane channel down here. It's a complicated phenotype, so I've separated them out. You're going to see the dynamics. This is a cyto D injection, which blocks assembly and disassembly. And you see the myosin's activated, and it tries to contract. But what happens is you see an instability in the cell connectivity. And you see the myosin structures start to come apart and separate. And you no longer get force transmission. And actually the mesoderm, which you see right here, is stuck on the surface. So it can't undergo the force propagation in order to invaginate the tissue. So what we think is happening here, based on that sort of piece of data and a lot of other data we've collected, is as these cells are dynamically contracting, individual cells will lose connections with each other. So you can imagine a bunch of people all holding hands. Maybe there's a lot of force. You'll have two people transiently let go. And in order to maintain a sort of stable network of connections over time, what has to happen is you have to have a way for cells to kind of grab back on each other. Otherwise, you have the chance of multiple cells losing connection and opening up a much more bigger hole than you would if you don't have this mechanism to rapidly reattach if there's damage. Yes, Vanessa. Yes, yeah. If you look at actin, you continually see these holes opening up and getting refilled.
But in the between membranes as well, like do you get gaps in the epithelium?
You don't get a gap in the epithelium. You get a gap on one side of the junction, and then the actin comes back and refills it, and then it reestablishes the balance.
Drug.
In the drug treatment, the holes are much more persistent, and you don't get the refilling. So we locked the system. It no longer has this sort of adaptive mechanism to sort of like reattach after you lose a connection. Yeah. We cannot do washout because we have to inject the drug into an egg that has a shell. So we haven't been able to do the washout. But we can have the embryo, we're imaging it, and like have the needle inside. And if we do an injection, you see this happen sort of immediately. So we can see a very acute effect. Yeah, Johannes.
So sometimes as you contract, so if I'm saying correctly, you deplete the actin from one side, so to deplete this whole. So do you think that the fact that you have this turnover is because you contract so much your actin that you start to depolymerize it, or there is an active signal that appears because the membrane is actually devoid of actin, and therefore you re-establish actin?
Yeah, we don't yet know. One interesting thing is when you get some of these dramatic detachments, you actually get Rho kinase assembling at the junction. So it's almost like there's a homeostatic way to sort of signal to Rho. But then it seems, I don't know if from the actin disassembly side, I don't know. The actin disassembly appears to be upregulated before apical constriction even starts. The mesoderm has lower levels of F-actin than any other tissue in the Drosophila embryo before the start of gastrulation, and that's snail dependent. So it seems like snail is somehow upregulating maybe the actin turnover so that it's primed and ready to sort of be dynamic. At least that's my model. Okay, one question I think is interesting, and I want to throw it out there in case anyone's interested in this, is, right, the cells are contracting kind of in a staggered fashion with each other. And I think one interesting question is, can this heterogeneity allow individual cells to kind of take turns contracting, right? You might think if they all try to contract at once, maybe they just fight against each other and are going to be unable to constrict. Whereas if you have a dynamic process, right, maybe here, this cell no longer has an opposing force. It's able to squinch in sort of its area, and then a neighbor will be able to then grab on all sides and then squinch in their area. So you get kind of cells taking turns. And this is something we haven't yet been able to prove, but it's kind of an attractive model, I think, for why you also require the dynamics in this system. Yeah.
So to contract the cell, you need the ectomyelin cytoskeleton to be anchored to the junction.
Yes.
Or you need to have high friction between the cytoskeleton and the apical membrane, so
Yep.
you can contract the cell. So from just what you say, I think this means that you think that basically the contraction will also likely come from the friction of the underlying cytoskeleton to the membrane and not only to the connection to the adherent junction. Because I don't see how a cell could contract its apex if either there is a high friction on between the cytoskeleton and the apical membrane, or if there is connection with the adherent junction. So
I think what I'm suggesting is you need high friction, but you also need sort of an imbalance. If there's high friction on both sides, then the junction might not move in. But if you have an imbalance and high friction on one side, then you'll cinch in. And then if the cell is able to stabilize its shape, then you give the neighbor an opportunity to kind of like pull in at another time. So they might take turns contracting in that way. But I agree, like if you get rid of the junctions, right, there's no friction. The myosin contracts great, but the cells do not apically constrict. And that's very clear. All right, so in this case, the cell dynamics results in kind of like a tissue level stability. Now I'm going to tell you about maybe a bit of a more undeveloped, I think, story, but I kind of want to get, it has crazy movies, so I decided to show it and see if anyone has ideas about it. So that's how do the forces and the activation of these cytoskeletal structures actually affect the signaling. And so a framework that's been developed to think about the excitability of Rho and the system is that you have an activator, in this case Rho A, that gets activated, and there's sort of autocatalytic activity, which leads to sort of a robust sort of burst of activity. And if there's delayed negative feedback through some inhibitor, whether it be a ROGAP or myosin phosphatase, that this would result in the type of pulses and waves that are observed. But then this inhibition could also be influenced by strain or the mechanics of the contraction itself. And so this is a model that was nicely modeled in the Staden paper from 2022. And so you get this autocatalytic activation of Rho, delayed recruitment of an inhibitor, and there's been a defined sort of GEF-GAP pair identified for this, and we extended that. The one that was first identified is this EC2 and RGA3. And I just want to point out that this kind of model is developed through the work of Akankshi Munjal and Tomal Laqui's lab, Bill Bement, Edmund Rowe, and also Zerhan Tan and Nikta Fakhri's lab. So I'll tell you one example of a developmental process where this type of excitability is actually activated during the process of morphogenesis. And I'm not going to go through the details too much because I only have 2 slides on this, but this is the Drosophila egg developing. There's support cells called nurse cells that shrink and empty their cytoplasm into the developing egg cell. And what Yasmin Imran Alsis and Jonathan Jackson discovered when they were in the lab is that halfway through this process, myosin goes from being isotropic across the cortex to developing this wave-like behavior that squeezes the cells. So this was the first example of waves that sort of came to my lab. What's that? Yeah, it looks like a washing machine. Yeah, so they're squeezing the cytoplasm, the remaining cytoplasm from those nurse cells into the egg cell. The egg cell's down here growing.
How do they squeeze it? They have holes?
What's that?
They have holes. How do they squeeze it?
It's like peristalsis.
But how do they dump the cytoplasm?
How does it exit the cells?
Oh, there are channels between all of the nurse cells up here, and there's channels between this layer of nurse cells and the egg. So there's basically incomplete cytokinetic sort of furrows. And so it's a cyst. They're all connected. And you get transport going all the way from the back here into the egg. And that gives the egg all the goodies it needs to develop into a healthy fly.
How can they rotate if they are linked? So it twists the channels or what?
We haven't seen that it has so much of an effect on the channels. It seems to be able to go around the channels. So.
Sorry.
Mm-hmm.
Do you see the directionality of this flow?
There is directionality.
There is directionality.
Yeah, and that is actually this is what we studied in this paper. I'm not getting into it, but it's the directionality is predicted by the radius of curvature of the nurse cells compared to the egg cell. So because this has a higher radius of curvature, it's predicted that there's greater pressure sort of with the same amount of tension and that you get directionality here.
I was wondering, of the nurse cell down, you have to have.
Yes.
Yeah, same thing? Or?
So it's okay. So I wasn't going to go into this, but I'll explain it. So these cells here have are smallest. So they have the highest pressure. So they squeeze, the greatest pressure differential is here to here initially. So you get an emptying first here to here, and you're not seeing that process. This is midway through the process, but you get a hierarchy in how it empties. Empties first here, then here, then here, then here. And the problem is there are big nuclei in these cells. So we think the waves, what they're doing is based on physics, you predict this will empty first, then this empties, then this empties. The problem is when this empties, you now have these big nuclei blocking the way for the other layers. So we think you need those active peristalsis to get cytoplasm sort of around the nucleus all the way to the oocyte. If you block the waves, you get it's like a clogged toilet. Like you basically get backflow and you get the cytoplasm going the wrong way. Yes.
at the much earlier stage, when they are determining which one of them is going to become the oocyte, they still maintain a connection between each one of these nerve cells. There are bridges between them.
Mhm.
At this stage, are these bridges gone?
The bridges stay till the end.
Cells between the nerve cells still intact.
Yep.
So then, when you're dumping things, then like it can enter the other ones as well, right? I mean, it's not
Yes. Yep. But the directionality is determined by kind of the shape hierarchy. Yeah, Sharid?
Yeah, so I was thinking, where does the force, retracting force come from? Does it come from the nerve cells themselves, or is the follicle cells around, which is sort of pulling these things back?
We don't think it's from the follicle cells. If we inhibit myosin in this in the germline, it dramatically slows down the transport. So we think the transport is being induced by sort of cortical tension in the germline system.
It's just total volume conservation. The oocyte is growing, so it's basically shrinking the nerve cells because everything else is there.
Yeah. Exactly. So if it shrinks here, it has to go somewhere. So it either goes here, or if it's backed up, then it goes the other way. Exactly. All right, but I didn't necessarily want to talk about this because what we did when we saw these waves, we saw that, I'll go back. Forgot this here. So I did genetics to identify a ROGAF and ROGAP that were involved in this. And what we found in this system is that both of these regulators basically were released from the nucleus. And when they were released, this is when you got the waves forming. So this suggested to us if we just increase the availability of both the GEF and the GAP, that you could stimulate this wave behavior. So we went back to our Drosophila embryo and the mesoderm to ask, could we basically induce a similar type of dynamic in the mesoderm? And it turned out we could. We co-overexpressed a different GEF and GAP, the two that were involved in the mesoderm. And what you can see is we actually, we changed the behavior of the pulses to be that of actomyosin waves. This is just color coding is showing you a temporal projection. So you can see that these things are really kind of propagating waves of myosin. This is an embryo that has cells still. So you're seeing the waves propagate from one side of the cell to the other. But Jonathan Jackson, when he was in the lab, discovered that he could actually get the waves earlier in development when there's just one big cell. And this allowed us to nicely characterize kind of the waves. So now we're not going to see multiple cells, but you're going to see one huge cell. Oh, that's too bad. I feel like the time resolution isn't as good here. Oh, see, there's some nice spiral waves of myosin. But what I want to get at, I'll try to play that once again. So you get these basically propagating waves of both RhoA activity and myosin. But what I want to show you is that what he did was he'd image an embryo, see these waves, he'd then take the embryo and inject it with latrunculin to show the effect of the cytoskeleton. And this is an embryo that's been injected with latrunculin. And what you see are the waves are still happening. Oh, let me go back. Okay, so initially you get these bands of myosin and RhoA, which you can kind of see there. When you inject latrunculin, you still get waves, but the structure of the waves is totally different. You get these spots of RhoA and myosin that are stationary, and you see the waves of activity move across those spots. So it's clear the cytoskeleton is really essential for the structure of not only the cytoskeleton, but also the signaling. Which we still don't understand. That's why I wanted to present this, because I'm hoping one of you can explain this to me. Yeah.
It's linked to, because I guess you have a transmission of information to make those waves, and the length scale at which you transmit must be dependent upon the length of the actin filament or the cross-linker between actin filaments. You have a length scale that must appear in terms of the. So I was wondering whether if you do slight layer depolymerization of actin, whether you will still reduce. I mean, maybe it's very and waiving the argument I'm giving that.
Yeah.
You should reduce the size of the wave, no, or the, at which it propagates, or.
But you actually increase the size of the wave, which I'll show in just a minute.
You increase the overall size, but the structure are very tiny now.
So here's an example where there's still some connectivity of the myosin, but you still see the rows and spots. And you see the myosin kind of pulling row spots and moving them around. So you'll have to take a look. This is very choppy, so you're not seeing the sort of continuity as well as you could, but I'm happy to show you. This is just to show that when latrunculin's injected, the period of the wave increases, the width increases, and the appearance dramatically changes. So rather than getting a nice band of propagating row and then myosin, which you can see the lag, you get these stationary spots that kind of like increase in activity and then go down. Yeah, Marty.
I guess when I was looking at the first movie of the mesoderm, I was thinking it looks a lot like the C. elegans embryo, but like mostly it flies. I feel like you don't really see these really big patches of myosin that are moving in this way. Or maybe I guess it depends on the context, but I guess I wonder, I guess because in this with inhibition, you really change the appearance. Does this kind of argue for differences in actin organization in worms versus flies, just like in general? Or do you feel like that's kind of too broad a swath? I guess, do these organisms use different tools sort of for wave?
I think they're arranged differently. I think they're the same tools. But I mean, like a lot of the types of behaviors, like there are also waves in C. elegans, I believe, during this process. So you see it's tuned slightly differently in different cases, but you can see how we can change the tuning even in the mesoderm cells just by overexpressing the GEF and the GAP. And the overall levels of myosin behave as you'd expect, right? You overexpress rho GEF, you drive rho A activation really high, but you don't get waves. But then if you express the GAP alongside it, you restore the levels of myosin, but you get this new dynamic behavior, the wave. So it's a complicated system. So just to summarize what I told you about how geometry and mechanics can gate signaling responses by cells. I told you about how cell dynamics results in sort of tissue level stability, and also how the cytoskeleton is important for organizing signaling in the context of the embryo. I acknowledged people as I went through the talk. I want to give a shout out again to the people I focused on their projects. So Soline Chenet did the work on cell division, geometry sort of, and the regulation of cell division axis. Nat Clark down here did the work on the germ band connected to the tissue folding. Jeannie Joden was a postdoc in my lab who did the work on the actin turnover. And finally, Jonathan Jackson, who's right here. Jonathan Jackson's kind of like Wilson on the Home Improvement series. You never get his full face in a photograph. So anyway, thank you very much, and I can take questions as you watch movies of waves.
Spots you saw in the lactate.
Yeah.
Are they anchored somewhere over the membrane?
I don't know. So that type of phenotype is also present in the mesoderm, I should say. And I don't understand it at all, and I would love to get at this. So if you inject the mesoderm cells with ROCK inhibitor or with latrunculin, the normal rho kinase structures disperse, and you get spots of just about everything in that pathway. So I don't know why actin is what it's doing to kind of restrain the rho pathway from forming these sort of aggregates, but it's a pretty universal feature, I think, in the early Drosophila embryo. And I don't know what is like organizing those little spots, but yeah, we're still trying to figure that out. I think it's kind of weird. You'd think you'd be like more diffuse or something like that if you got rid of actin, but yeah, we get spots, and they're pretty consistent size too.
Do you think that the normal bands are made of those tiny spots that are organized in a different way then? Or do you think it's a completely different structure?
They look pretty different. Yeah, I don't, because if we could see the spots coming together, right, you'd see them coalesce. So we see that in the mesoderm normally. But in this case, it's really a very diffuse, continuous sort of band that propagates. Yeah, but I mean, maybe with higher resolution imaging, we could tease it apart better. Yeah, Sean?
Do you think that you would see the actin cortex holes in other scenarios other than the one that you showed? Just because I'm thinking that it's usually thought that there's always a surface tension caused by the cortex, and to some extent, if you didn't have those actin turnover dynamics, maybe there would be little holes, but they've gone unnoticed, and your system's sort of able to exacerbate them. Whole nature of the dynamic.
I think the forming of the holes is a fundamental feature of actomyosin. This has been modeled. Michael Mack from Yale had a nice modeling paper on this, where when you get contraction, right? If you just think about it, you get myosin contracting actin, right? It's going to cluster the actin filaments. And if you're kind of pulling actin filaments in from and concentrating them somewhere, you're losing them from somewhere else. So they've theoretically treated this with an agent-based model to show that it's the case. I think the phenotype where you get disconnections when you inject things like Cyto D or disrupt turnover is fundamental. That's seen in fibroblasts. It's seen in fission yeast during cytokinesis. It seems like the turnover is essential to maintain connectivity of these structures, both in theory and in all of those experimental systems. So I think, whether or not you can see the holes as nicely, we might have a system where we were able to see that a little bit better. But in each of those cases, right, in fission yeast, if you take a cofilin mutant, what happens is you get all the nodes of myosin collapsing into a big node rather than forming a nice continuous cytokinetic ring. And that's the work of Tom Pollard. So, yeah.
I guess you have looked at the ROGAF and ROGAP in this context.
We've looked at Rho. We've stained the gap. It's a little harder to track the gap.
But you think that they will move with the wave, or the gap will somehow just based on interaction with Rho and Rho interaction with Rho kinase.
Yeah.
Do you think that they will be moving with the wave and it's how they control the wave pattern, or this interaction is not important?
Yeah. In these guys here, the Rho leads the myosin by 3 seconds. So you can see kind of the direction of the signaling. We haven't been able to do this with ROGAF2, sadly. It's just our ROGAF2 GFP is too faint to really detect it. And with the C gap, we haven't been able to see it live, but we've done staining, and it looks like C gap might be slightly behind the wavefront, as you'd expect maybe from an inhibitor. So it kind of overlaps with the F-actin.
And do you have any idea? I mean, how the gap could be recruited in response? Is it the concentration of the actin that recruited, or the
possible, but we don't have any. So the C gap is a gap domain and nothing else. So there's nothing there that would suggest that it's going to bind to F-actin. But in the ventral furrow, in the mesoderm cells, it seems to be fairly cytoplasmic. And if it localizes anywhere, it localizes to the lateral edges of the cells. So how C gap is getting anywhere is still a mystery. Yeah, Olivier.
So the reason why you see these waves is because this is a very large cell, I suppose.
It just makes it nicer, yeah. But you can see the waves if in small cells, right? That's in this case, right? A cell is like maybe like that big, and you're seeing the waves propagate in the small cells.
And you see the spirals, small cells.
Well, because the waves are in different cells, then you don't see them. They don't cross over. So So you can't see the excitability, right? With the big cell, right, you can see the waves annihilate, you can see spiral waves, you can see all kind of the behaviors you'd expect from an excitable system.
Are there any other examples of such waves of, with the same system with myosin?
Yeah, certainly. There's the starfish and xenopus. Yeah.
The waves on the cortical surface of the xenopus egg.
Exactly. Here's a movie from Bill Bement's lab down here. This is row. So you're seeing the row kind of propagate from one side to the other. And Bill Bement has shown if you overexpress a GEF in xenopus and in starfish, you get the types of behaviors that I showed as well. Yeah, exactly. Mm-hmm.
So saw them just increasing the temperature.
Huh.
So I'm
Yeah.
thinking that might have something to do with actin turnover.
Okay. Yeah, for sure. Actin turnover is very sensitive to that.
Exactly.
Yeah.
Not trying to do anything of the kind, but somehow, the microscope was a little bit warmer that day. And
Yep.
we saw the spirals come out really just quickly, and then they disappeared.
Yeah.
Yeah. So that might be something.
Yeah. That's a good point.